You're listening to DNA Today, where we cover new advances in the world of genetics. I'd like to introduce and thank Dr. Rachel O'Neill for being on the program today. Welcome.
Thank you.
Dr. Rachel O'Neill received her BA from the University of Texas and her PhD from La Trobe University, Australia. She's now the Director of Center for Genomic Innovation, and she's also a professor and head of genetics and genomics in the Department of Molecular and Cell Biology. All very impressive.
Thanks.
So I want to start by asking how you got interested in genetics, how you started pursuing genetics as a career.
That's actually a funny story because I didn't really know I was interested in genetics until I hit graduate school. And then looking back, I'd always been interested in genetics. And it was just this very weird denial that I must
have been. It's like that hindsight, like, I've always been into it.
Very much so. And so what really triggered it for me was when my mother sent me all of my high school documents after I moved back from Australia. So this is long after my PhD and postdocs and whatnot. And in there was my freshman biology term paper, which was called From Genotype to Phenotype. And it was about UV irradiating bacteria, which was my project. And at that moment I was like, I guess I kind of always dig this topic. But just didn't really know it.
Yeah, it wasn't in the forefront where you're just like, this is what I want to do.
Yeah, it was always the thing I thought, wow, that's really cool. If I heard a science story, I really gravitated toward those kind of stories. I thought those were the neat ones. When Gattaca came out, I thought it was an awesome movie. And it's like looking back, like, that was obvious, and I just didn't get it. But hindsight is 20/20.
And now you're fully in genetics and just busy with everything that you're doing. So
every minute.
What do you do in your role as the director of the Center for Genome Innovation?
So we do a lot of different things. So that center is actually a rebirth of a center that was started over a decade ago by Linda Strausbaugh, who recently retired, who is an amazing geneticist. And I learned a lot from her on more than just science, but engagement and making sure that people were involved. And so we are continuing in that goal with the center. We are under the Institute for Systems Genomics. So we have an education and outreach mission where we are making sure that students are coming in and learning the technology. They're the ones that can actually take the technology to the next level. But we're also facilitating scientists across the campus and hopefully broader than that in working together, obtaining new techniques and new technologies that maybe they didn't have access to before, and putting scientists together into bigger, more collaborative projects.
Moving them on to that next phase.
Yeah, very much so. Very, very much so. And one of the classic examples I use is one that I was lucky enough to be involved in. It was a marine science professor who was working on climate change and using SALP, which is an organism that she actually studied. And she wanted to add a genomics component to that and understanding how that could be indicators of climate change in Antarctica. And so they came to us, and we had her student downstairs and trained her to do some of that genomics stuff. And it turned into a really interesting collaboration because I learned about a whole new organism that I didn't even know much about at all.
So when people are being incorporated into these projects, is it very much like learning on the job, training on the job, or is it a lot of applying your prior knowledge and education to that specific
all of the above.
Research?
All of the above. I mean, we have students and scientists that come in completely naive on what genetics and genomics is, and some that are absolute experts. And we want to help everybody at whatever level they need. So it's absolutely all over the place.
And as a professor, you're very much in the education and teaching aspect of genetics and everything. What classes do you teach at UConn?
So I teach 3 classes right now that I actually love teaching, which you don't hear many people say.
No, you don't. It's always great when you hear professors actually loving what they do.
I co-teach all of them, and the person I co-teach with all of them with is Mike O'Neill, and we collaborate as well. And we've developed 3 really interesting classes. One is a freshman honors non-majors course. So this is for anybody on campus. You don't have to be a scientist. We end up with a lot of scientists in it, but it's a general education course. It's an honors core course, and it's called the Genetics Revolution in Contemporary Culture. And so our goal with that class was to try to cover the fundamentals of genetics and genomics, but using popular culture as a way to do it. So using movies, using books, art, newspaper articles, humor, Colbert Report, for example. We just showed a clip from that on Schmeat. We read the book Oryx and Crake, which is a Margaret Atwood book about genetic engineering gone wrong. And then we actually have a mock trial about it. So we really touch on the ethics and the law. But, fundamentally, we firmly believe that genetics and genomics is an area that touches every single person at some point in their lives. They're, giving their genetic information away when they go to a doctor's office and they fill in their family history.
It keeps following them,
so
yes,
at
every doctor's appointment.
Forever, right, exactly. And we think that, theater, art, movies have really tapped into genetics and at least the modern fear of it.
Yeah, even in sci-fi, you mentioned Gattaca before, that is the prime example.
Yeah, there's so many really bad, horrible.
And there's some good ones, but there's a lot of bad. Yes.
Yeah, there's some really good ones too, right? So, and you always see, genetic engineering is thrown in. And Spider-Man is a classic example, right? So in the 50s, when Spider-Man came out, it was radiation. It was a radioactive spider that bit him, and all of a sudden he had Spidey powers. And now it's in both recent iterations, it was a genetically modified spider, right? So genetic engineering has, geneticists we're all crazy mad scientists, but it's what is embedded in our culture today.
Definitely, yeah. So I want to get into kind of what you do at your lab. You use molecular genetic and cytogenetics techniques to study centromere determinants. Can you break down for our listeners what this means?
Yeah, that's besides just a mouthful.
Yeah, it really is. Yeah, I struggled through that.
Fair enough. Yeah, so the best way to think about it is if you're a diploid organism, your cell has to divide. And every cell division, whether it's somatically or in your germline, needs to be accurate, right? And so there's an architectural region of the chromosome called the centromere. And people probably have seen it when they've seen pictures drawn, even grossly, of a chromosome. It looks like a big X.
Yes.
Right? So the centromere is that spot right in the middle. And so it's very specific and very architecturally defined, if you will. But it's also absolutely critical. If your centromere doesn't work, your spindles can't bind anything and your cells don't divide properly, or you lose chromosomes, or you get chromosome damage, chromosome breaks if you have 2 centromeres on a chromosome, for example. And these are some of the massive scale rearrangements we see in things like cancer. So we're trying to understand the fundamental processes of how centromeres function and how they've evolved.
Because in high school biology, they say, yeah, this is what it is, and people are like,
what
does it do? And you're kind of like, well, I'm not really sure.
In fact, in high school biology, they also tell you it's heterochromatin, it's transcriptionally inert, it doesn't do anything. And all of those are not true. It turns out recently they discovered satellite overexpression is a great biomarker for prostate cancer. Right, so these are really important regions of the genome, and not only do we not know how they function, we don't know how they evolve. And there have been some amazing studies done recently on a variety of different organisms, and I think the gibbon genome is a really great, interesting case where they can really finally tap into the types of rearrangements that have happened between these species. And centromeres are highly active in that and really important. And another thing that's interesting is that we haven't actually sequenced a full human centromere yet.
Really?
So every time they say the genome's all done, they're actually
That's what I was going to say. I was like, I've heard that it was all sequenced.
And then when you see those T-shirts that say, you're 98% chimp,
right?
And while you might like to use that description for ex-boyfriends, it does not actually apply. Because if we just take the sex chromosomes, for example, human population can vary in satellite content for 10 megabases. That's a huge amount of DNA variation that even you and I may have that's different. Now multiply that times 46 chromosomes. So the potential for variation is actually a lot. And so we really don't know what variation exists in the human population, and we don't really know how that's defined between species even. What we do know is that it's very rapidly evolving. It's one of the fastest evolving regions of the genome. But functionally, it's incredibly conserved. So those are some really interesting paradoxes and some really interesting mysteries of biology, and that's kind of what I'm interested in.
Certainly, yeah. In 2011, your lab, including Dr. Mark Longo, he actually has one of those shirts you're referring to, the 98% chimp. Is that why you brought it up? Yeah. I was going to say, he wore that to lecture once, and I was like, that's a cool shirt, but now that I know it's not too accurate, I don't know if I'm going to get one. But
it's all content.
So he was a graduate student at the time, and now he's an assistant professor in residence. As I said, he's one of my professors, actually. So you researched the contamination of DNA samples within human DNA.
Right.
And you sought out to find how many databases contained human contamination, which it doesn't even occur to me, and I'm sure a lot of other people.
Well, it's funny. So it didn't occur to us either. So this was a story of taking a very depressing day where a student realized that what he thought was an ultra-conserved repeated element that was, it was in all mammals. And the more databases he looked at, the more he could find this. He could find it in zebrafish. And then he, just kept going further and further out, this phylogenetic tree, and the sequence wasn't really changing much. And at that point, we realized that's probably not an ultra-conserved sequence, but a human contamination.
That it happened in all of these.
That it's in the databases. It's not actually in these organisms.
Which I imagine has huge implications. If there's contamination in all of these databases, how can we...
Right. And I think, everybody knows that there is. Everybody knows there's, because if you think about genome assemblies, sequencing technology today is not capable of jumping very far in sequence, right? So the standard technologies can get 500 to 1,000 bases. Some of the higher scale nanopore sequencing type technologies can get 10, 20, 30,000 bases. But the reference assembly is whole chromosomes, right? So this is a consensus sequence that's assembled completely computationally, right? This is an artificial construct that exists in a computer that hopefully replicates what exists in human cells.
But not necessarily.
So when you're putting these assemblies together, and this is why the centromeres haven't been done, when these assemblers hit these highly repeated regions, they have a very hard time placing where the DNA could go, especially if there are 1,000 copies of a sequence that are exactly the same in the genome on different locations. So the assemblers sometimes miss sifting through and pulling out human sequences. And that's certainly what had happened in a lot of these reference assemblies. And a lot of people sort of knew this happened, and I don't think anybody knew how pervasive it was, and that it had actually gone into final assemblies rather than just rough assemblies. And what we hope that study did, besides just upset a lot of people, was just make people realize, these are computational constructs, and we're still tweaking them, and we still have a way to go.
Nothing's 100% accurate.
Nothing's 100%. But I think, for us, it wasn't more of a comment on those assemblies, which are still amazing considering where we were even 10 years ago, is the concept of personalized medicine. So if we can get all these assemblies and they have human contamination in them, what happens when we start doing personal genome sequencing? How do we distinguish variants from the sequencer?
And that becomes, if you're looking at genetic diseases someone might have, it's going to affect that result.
So the idea behind that paper was more, not don't do it, but if you're going to do it, treat it like forensics. Treat it carefully and with some modicum of caution when you're preparing your samples, you're making your libraries, etc.
Definitely. So another main focus of your lab is studying species-specific placenta development and evolution. Can you talk about what you're currently doing to study this?
Yeah, so we started working on this fish species. It's a desert top minnow, and it's got a bunch of species in the complex, actually. And we didn't discover this. Somebody else discovered this years and years and years ago. It's an interesting story of why we started working on these fish. So when we first moved to UConn, a professor in EEB had just retired, Jack Schultz. And he had 3,000 fish.
And you're like, guess what we're studying?
And he's like, here, take all these fish. They're really cool. And we're, okay. And we started looking at some of the biology of the fish, and they did some really amazing things with their chromosomes. For example, some of them are clonal. They actually clone themselves in reproduction, and that's classic work that pioneered here from UConn. But some of these species have also evolved a placenta, and it's a placenta that's analogous to a mammalian placenta. And in fact, it turns out mammals aren't the only thing that develop placentas. There's a fish.
That's
spider,
shocking, yeah.
There's a beetle. I mean, there's all sorts of random species that have converged on this very critical phenotype. And it's a phenotype that's interesting because it can also become a a place where parental genomes and embryonic genomes can actually come into contact and be in conflict. And that's the classic case, at least in the mammalian placenta. And so the question that we were asking was, do these types of principles and conflict exist in derivative placentas that have sort of evolved independently? And what types of genes are involved? So, for example, in mammalian placentation, retroviruses are very important. And they've been co-opted in a very species-specific way. And it's a very fast-evolving organ. So how does this look in a completely independently derived system like the fish? And so that's sort of what we've been looking at.
She's referring to
a picture.
Pictures and yes.
So it's derived from the pericardial sac. So, but it does maternal fetal exchange, just like the human placenta.
And so it's very similar to mammalian placenta.
Absolutely.
Wow, that's
physiologically and functionally,
and this is happening, you're saying, through retroviruses?
Well, that's what we're testing.
Okay. We've
actually identified some transcripts that are pretty interesting. In mammals, viruses have been co-opted. Syncytin, for example, is a great example of this. It's a human protein that's involved in the early stages of placentation in forming the syncytium trophoblast cells. And it's actually a fusion gene that has incorporated an envelope protein from a virus.
Wow. Very interesting. So we mentioned that Dr. Mark Longo worked in your lab previously. Yeah. Who's currently working in your lab, and what's their level of education and experience?
So I have students that sort of run the whole gamut of education and experience. I have a rotation student that's been in my lab for just a couple of weeks. She's just starting her PhD. I've got PhD students that are in their 4th and 5th and 6th year. I've got some PhD students in their second and third year. Got some master's students that are just starting out. I've got some undergrads.
Wow.
I've got some
quite a range.
Yeah, it's quite a range, actually. Yeah. And one of the undergrads actually started his freshman year. So I've had students that have been undergrads that stay for grad school. I've had students that have been undergrads that go off and do amazing things at other places. It runs the gamut. I've also got postdocs. I've got technicians, and I've got senior scientists and faculty.
So if someone is interested in working at your lab, is there a process for applying, or how do they come in contact with you?
I think just coming in contact with me, just writing and saying, hey, I'm really interested in what you do, and I'd like to talk to you and learn more. I think I'm a big proponent for students that are interested in at least trying out science to get their feet wet, because it's just as important to know if you love doing bench work as it is to know that you don't.
Yes. Figure that out now.
Right. And that's kind of why you're here, right? This is the exploratory period of your life, right? So you really want to take those opportunities when you can. And at the end of the day, if the biological question doesn't drive you, nothing will. So one of the things that really hits me when a student contacts me, whether they write me by email or come see me, is that they know something about what I do, whether they've just read it online or heard somebody else talk about it, but it's piqued their interest. And they may not know much about it, and I expect they probably don't, right?
Even reading through it, it can be very dense.
It's a very specialized, narrow field, right? So, but just to be excited about the same types of questions. And those are the kind of people that I'm looking for. Those are the kind of people that come into the lab and flourish.
Certainly, yes. You have to have that drive in order to be going through all of these kind of somewhat tedious steps, I imagine.
I mean, it's drive, but it's also a really innate curiosity and a little bit of stubbornness. And a lot of stuff doesn't work.
I'm sure stubbornness can go a long way in this field, certainly.
A lot of stuff doesn't work. So you really want to be dogged about it.
Definitely. So scoping out from your lab, how is UConn on a larger scale contributing to the field of genetics?
I think it's contributing greatly, and I think it has for a very, very long time. I think it's... If you look in any genetics textbook, there's a very, very famous photograph that was taken on the football fields of people in their little UConn t-shirts, and it's a blue and white t-shirt photograph that just measures height. And it's a classic genetics textbook. Well, that was taken by Linda Strasbach here at UConn. Right? So I think there have been a lot of famous geneticists and genomicists that have come through UConn, but even today, there's been a new resurgence of genomics, especially with Jax coming into the state, some new hires, some people that have been around for a while and are really picking up genomics and genetics tools. We see it. It is not department restricted. We see it campus-wide. And in fact, across multiple campuses. So I think it's a really big and exciting time. I'm a little biased because I'm in it.
That's okay.
But, the diversity of things that I'm hearing people do is really cool. I have to say, it's really cool, and it runs. It's so big and it's so broad.
And you're definitely right about having the different departments all in genetics, because
absolutely.
there are a lot of departments that even have somewhat of a genetic component or even concentration throughout UConn.
Yeah. Very much so. I mean, we'll see behavioral psychologists that are tapping into doing some genetics and genomics.
Yeah, there's even some psych classes that are genetics. It has it right in the title there at UConn.
Exactly. There's even classes in the law school. I mean,
yeah.
It's one of those fields that really does tap into so many different disciplines.
And UConn also has a new partnership with Jackson Laboratories. We both went to the grand opening of Jackson Laboratories for genomic medicine a few weeks ago on October 8th. Yes. And that was a very exciting day.
It was very exciting. It was a beautiful facility.
It really is.
It was really nice to see it. It was really nice to see all the scientists that were there. It was really exciting. It was an exciting time. It's been exciting. It's been exciting since day one.
It has. I remember being, I believe I was a junior in high school, and I found out that the partnership was happening and that Jackson was being built in Farmington, Connecticut, and just that exciting day of things are really picking up in, the bio field, especially genetic field within Connecticut.
Yeah, very
and
much
definitely
so.
UConn having a big impact.
And I think UConn and Connecticut in general has had a really big stamp on that field. I mean, we've had 2 companies that have started in Connecticut. They were pioneers in next-gen sequencing, for example. And having Jax GM decide that, Connecticut was the place it was going to put Jax GM was wonderful for everybody involved, because it was a way to really bridge all of the universities and all of these companies in ways that we had already been doing, but not vigorously enough. It really just
really enhancing those relationships.
Yeah, it really did.
Yeah, and then anybody that works at Jackson might have the opportunity to go up to Bar Harbor, and that's pretty cool in itself.
Yeah,
it really is beautiful. The mansion there, I've seen it from afar, and it is quite nice.
I have to agree.
Yes. Is there any presentations or new research that really struck you from the conference?
I think, yeah. I mean, I think it was just the scale. And I think that it sort of, I know this being in the field, but it's really nice to hear the different angles that people are taking on this big data approach, not just looking at one patient and one chromosome locus, but looking at hundreds or thousands of patients and the whole genome. And it doesn't matter whether it was disease or a particular biological pathway or developing a model or a tool. I think that, to me, overall, was the amazing thing.
Yeah, and just the vibes there were just really great. Everyone was just very excited to be there.
Excited. Yeah, and I think that it's not to be understated. It's just really cool when you meet other people that think what you do is also cool.
Yeah, other people, you're like, genetics, they're like, oh, that's interesting. Moving on. Yeah.
You don't like thinking that what you do is not meaningful or esoteric in any way, so it's kind of neat.
So I want to end with what you see the hot topics in genetics being. Where's genetics headed? What's the next big step for genetics?
The next big step for genetics, that's a really big question.
It is. I need the answer right now.
I have been put on the spot. I think there are a couple. I think one...
You're going to cheat, aren't you?
I'm going to totally cheat. What I'm going to do is I'm going to talk my way around the answer, which is what scientists do really well.
I'll start getting used to that, but yes.
I think epigenetics, and I think that it's been a field that's been around for decades, but understanding the contribution of non-coding RNA and epigenetic signals in different susceptibility phenotypes, whether it be cancer or addiction, transgenerational inheritance of some of those signals, so the information that goes well above and beyond just that GACT and how that is manifest globally.
Everything, how that's regulated and all that comes
And I think it really is that, and then, a sister to that is how to tweak it. And that's all of this new editing technology that's coming.
Well, thank you so much for being on the program. I want to plug your website, roneal.lab.uconn.edu. You can find out a lot of information about what we talked about your lab and you as a professor and
Indeed.
Geneticist and all that encompasses that.
Yeah, science rocks.
It certainly does. All right, thank you so much.
No problem. Thank you.
That wraps up today's episode of DNA Today. Tune in next week, Monday at 1 p.m. on WHUS.org or 91.7 FM to learn more about genetics. DNApodcast.com is full of content, including all episode broadcasted, as well as mini lessons on genetics and resources to expand your knowledge in genetics. And I really highly encourage people to check out the website because I have so much more on there that I can't share on air. So it's a great resource to go to, and if you have any suggestions on what I should add on there, you can email in at info@dnapodcast.com. I love hearing feedback. If there's anything you'd like me to talk about on air, a certain genetic disease or news story, I'd love to be able to do that, and even include your own opinions on it and your own kind of insight, and I would love to also share that on air. You can also follow on Twitter at DNA Podcast, and you can follow me on Twitter at Kira Dineen, KIRADINEEN. Thanks for listening, and join me next week, Monday at 1 p.m. to learn and discover new advances in the world of genetics.